Hi there.
Hello,
Hi.
Dr. Seneff. How are you doing? I was
I'm good.
all worried that we're having technical issues. We always kind of joke about this because everything that I'm doing relies on technology, my music and my animation.
I know.
Everything starts to glitch, and we actually call it glitchosate after glyphosate.
Glyphosate. I love that. Glyphosate. That's beautiful.
Well, as you can see behind me, this
Look at those.
understanding I've got
Wow.
so there's deuterium and there's hydrogen. The doll maker didn't quite get that I wanted hydrogen half the size of deuterium.
Whoops.
But then we've got glyphosate right there.
I see glyphosate. That's
Yeah.
lovely.
And then
Wow.
some vitamins and minerals behind there. So
Oh, that's funny. You're gonna have to make a new hydrogen.
I definitely. It's interesting. You know how long I've been working on this, and the really cool part is that I knew there'd be no overnight success. What I'm saying
Right.
is it's kind of out there in certain ways, but the world has come so much closer to what I'm doing and what you're doing through COVID-19, that all that
I
nonsense.
know that
So
really accelerated things, and I think I want to get deuterium out. That's why I'm so glad that I tell people if they ask me if I can do a Zoom interaction, for a podcast interview, I say no, not unless it's deuterium. It has to be deuterium in there somewhere, or I
well,
won't do it. So
yeah, that's great. Well, I just finished watching the podcast that you did. It's great, but I've been following this. It's interesting. So about 7 years ago, when I first met you, I was actually sharing workspace with Laszlo Boros, which is
I know you were. That is so ironic.
it's
It's a
crazy.
small world.
I wish I would have introduced you. I've always been kind of shy about this because, I'm just like a clinical dentist and I'm reaching out to these researchers and, I wish I would have introduced you long ago, but it's interesting that you found each other. So,
I know it really is, and it's so exciting. It's funny because I've had two major points in my career the last 15, 20 years. One was when I heard Don Huber talk about glyphosate, 2012, and the second was December 2019, right before COVID hit, when Laszlo basically sent me an email. He said, "I loved your article. By the way, deuterium." Basically, that was the message in the email. I'm like, oh, deuterium. I didn't know
right,
anything about that. I knew what it was, but I didn't realize it had such a significant role in biology.
right,
And that's just been a wild, just been wild, really wild. And now
right.
I'm really on fire with it. I've gotten to a new point with it, and it's kind of driving me nuts. Like I can't even keep up on the email because I'm so busy reading papers.
I totally get it. It's very interesting because for me, I've done a lot of podcasts on this, but for me it was hearing Laszlo Boros speak. And this was obvious. It was very obvious to me how important this topic was. But again, I was kind of shy about pushing it just because, I don't I have a doctorate of science through my dental degree, but I don't I was nervous about this, but it's a big deal. And I loved it when you sent me that last email. You're like, this is like the theory of everything, because
Yeah.
that's how I felt about it for a really long time. Right.
Yeah, I think deuterium is the number one factor
Yeah.
messing up our biology today. And it's because of the chemicals that are messing up the deuterium. So it's just really, really
Well,
interesting.
so interesting that mixture. So I met you specifically to speak about glyphosate. And if anyone wants to go back and look at our podcast, we've done a few. I'm going to be relaunching them, rebranding them. But what I really wanted to do with you, just because seeing you go from, it's just a little context. When I read your paper, is endothelial nitric oxide synthase a moonlighting enzyme? That kind of blew my mind. And so I want this podcast. I know that you and I both really want to get into the science geeky part, but I want to keep this podcast at a specific level because I want to use this particular podcast as the introduction to a series that I've done, a podcast I've done on deuterium. I have some really cool guests, and each one of them has a very different aspect of deuterium of their as their focus. And I wanted to, because I think that your information on so many levels is so profound. I wanted to do this podcast with you. So I'm going to slow you down and I want to take you through what I think that a way I think people will be interested in hearing about this. Because when we say deuterium, the first thing is they like, oh, you think Deuteronomy, which is fascinating. But some people will say, oh, that isn't that, heavy water. And to anyone watching this video, we're going to go through all the details, but if it's confusing, there's lots of other podcasts that I've done And we've done, and there's, you know, I'm going to be doing a whole series on this. So I want to bring out this information in a specific way so that we get most people to stick with it. There's going to be some real life information that's going to be obvious that's going to come from this. But what I like about this topic, and a lot of your topics are, when we look at scientific studies, who's doing them? Who's paying for them? And what are they going to show us? What I like about the stuff that we're talking about, this is quantum biology or quantum physics, and you can't argue with an extra neutron. I kind of like to say that. It's like, you can think whatever you want, but ultimately you can't fool Mother Nature. You know, that's one of my catchphrases. So first thing I'd like to do is let you introduce yourself and give people some context. You're a very unusual person who's bringing together a lot of different education and background. So please introduce yourself and just give us the elevator pitch of who are you and why should we care? And I think we should care. I think that you've got some incredible information out there.
Thank you. Yeah, well, so my entire career has been at MIT. I have an undergraduate degree from MIT in biology. And then I have 3 other degrees, a master's, a PhD, and an engineering degree, also all from MIT in electrical engineering and computer science. So it's a mixture of biology, computer science, and electrical engineering, which has served me really well, I would say. And most of my career I was writing computer code to help computers interact with humans using natural speech. That was kind of my main career path for most of my career. And it wasn't until about 2007 was when I got worried about autism. I saw the rates going up every year. I got really nervous, and I love children. I want them to be healthy. I was very concerned. I thought, we've got to figure out what's causing this, because it's not genetic. There's genetic predisposition, and kids who have certain genes that are defective are going to be more easily affected by the toxic exposures. But what are the exposures? I didn't know. I wanted to find out. And I managed to make a nice switch in my career path and keep my funding, which was really, really wonderful. So I'm funded by the same computer company that funded me to develop dialogue games to help an English-speaking person learn Chinese. So it's a very big change in my career path in 2007, where I transitioned into this, looking for correlations between toxic chemical exposures and disease. And so for 5 years, I pored over the autism literature, getting a broader understanding of what autism is. Extremely complicated disorder with many comorbidities associated with these brain dysfunction that's manifested as social awkwardness, and many of them can't speak, you know, and severe disabilities in many cases. So the brain is obviously being affected. I've learned the gut is being affected, and it's a gut-brain axis, and that's contributing in all of that. So I was aware of all of this, looking for things and looking at correlations and not finding anything really for 5 years. And it wasn't until I happened to be at a conference, Professor Don Huber was speaking about glyphosate. I didn't know what glyphosate was, but I thought, well, here's a chemical, I better find out about it. And when I realized it was Roundup, I was very interested, because I knew Roundup was pretty pervasive in our environment, considered to be perfectly safe by the government, completely unregulated. And he showed, you know, how Roundup affects the soil bacteria, the gut bacteria, chelating minerals, you know, causing damage to liver enzymes, lots of things. And all of those things fit very well with autism. I knew that because I knew about autism. I thought, oh my God, this is it. It was really an epiphany for me. And I went back and just read everything I could about glyphosate. The rest is history. Eventually, I wrote this book, which I have over here.
Please, yeah, let's see
Toxic Legacy.
Yes, that's some pretty heavy reading, but it's very well done.
Yeah,
this is for anyone who doesn't know what glyphosate is. You know, we'll go over it briefly, but the book is incredible, because this is something that's so difficult to get your mind around. You can't smell it, you can't see it, you can't taste it, but it's probably almost everywhere. So, all right.
It is, yeah. And in fact, you know, Zen Honeycutt's a friend of mine, and she's the founder of Moms Across America. She's doing great work, and she's done studies on school lunches. So she got her friends, she has a network, and she got her network to contribute samples of school lunches in the grade schools, you know, the high schools, and send them off to get tested for glyphosate. And then she's just recently done the same thing again with fast food. So she's got samples of food from the 20 top fast food restaurants. Really? 100% of them tested positive for glyphosate.
Okay. And what type of, just to interrupt, what type of amounts are we talking? So, because I mean, it makes sense. We're growing GMO corn, grain, soy, and someone's got to be eating it. It's got to be there. So I would assume it'd be, in all the fast food. Was it higher levels than we have seen? To my knowledge, the levels are going up every year.
Yes, that's generally true. And of course, the US actually has a lot more than other countries. There's an interesting paper on honey. They sampled honeys from around the world, different countries, honey, and they did a big chart. They had this big graph of the levels of glyphosate in the different honeys, and they had them organized by country. And you saw this big part in the middle that was US, all with the high levels. It was very clear. You could almost draw a circle around where do you think it's US, and you'd be right just by picking the ones that have the high levels of, and almost, most of the honeys were contaminated from the US.
Right.
And
And
that
everyone's,
shows the bees are getting exposed, and the bees, of course, are dying of bee colony collapse syndrome, which I think is glyphosate's a huge contributor to that, along with the insecticides.
yeah. All right. Well, so I want to get into this just so we can talk about, glyphosate's scary. It's very hard to avoid. But we talked about this before. Buying certified organic is your best way, but what's dangerous about glyphosate is that it's sprayed on crops to desiccate them and to dry them out. So it's in a lot of different things. So maybe just give just brief what foods should people avoid where we use a lot of glyphosate.
Yeah, it's kind of surprising. And actually my friend shoot. Lost his name. Anyway, there's an Indian friend from
I know, yeah,
from Canada.
I know. Yes, I interviewed him. He's a really cool guy.
Yeah, did you lose his name? Also, I know his name, but it's not.
It's going to come to me too.
Moment.
So hold on, it's going to come to me.
I have a little bit of a problem with. I have a problem with names, so I'm sorry about that.
No,
but
no problem.
Anyway, he got the Canadian government. He's Indian descent, but he's a Canadian citizen. He got the Canadian government to test for levels of glyphosate in Tony Mitra. Sorry, I often get the name indirectly. When I start talking about him, I get his name, Tony Mitra. He got the Canadian government to test over 8,000 different food samples for glyphosate. The U.S. government has tested almost nothing. As far as I know, only soy, only in 1 year, 96% of the samples were positive. And, so it's clear that we have a problem. But Canada, over 8,000 samples, Canadian origin, coming from the US, coming from Mexico, coming from Europe. They did, imports. And then he wrote a book called Toxic Poison Foods of North America.
That's right.
Foods of North America.
And I read that. It's a very difficult book to get through. But the basic idea is that you have to understand, and this is, to anyone watching this, is not a surprise that our bigger government agencies are captured. And, if you don't test for something that should be in everything, then you don't have to think about it. And there's so much money involved in this. People will think, oh, Monsanto, there was a judgment against them. And there was, but they're still making this product because it's still very profitable. And I always wonder, we've talked about this before, whether someone is intentionally poisoning the world or whether it's just profit driven. Because to me, it seems like the company Monsanto, now Bayer, they don't need the money. They don't need the money.
It is amazing that they are willing to poison the world, really, because and the thing that's exciting is that the papers are coming out faster and faster. There's exponential growth in the number of papers coming out that are showing glyphosate's true colors, and very exciting. Just last few months, I believe, there's a paper that came out, I think, just a few days ago out of Brazil that showed that the growth of the soy and the places where the soy crops are being prepared, grown in Brazil, have higher incidences of childhood leukemia. And childhood leukemia has been going up every year in the United States. And then there's another paper on rats that exposed, and this is from Ramazzini Institute I think they're in Italy. They're doing great work, and they're doing a lot of studying on glyphosate exposure to rats and looking for things. And they have found an incredible result with extremely significant correlations between glyphosate exposure at low levels, below regulatory limits, even way below regulatory limits, causing leukemia in rats at a young age. So this is basically childhood leukemia in the rats. And they have just come out with news on that, and it's being picked up by a bunch of news media, mostly, of course, alternative news media are writing articles about it, because it's really the combination of that plus the childhood leukemia in Brazil, and then the fact that leukemia is going up in the US. I think there's going to be leukemia lawsuits, childhood leukemia lawsuits against Bayer. And also there's a new lawsuit in I think there's a new lawsuit in Missouri, St. Louis, Missouri, and also in Pennsylvania, two new ones. There were the three out of California for non-Hodgkin's lymphoma. There were three in rapid succession with jury trial and huge awards. And then now there's two more, one in St. Louis, Missouri, which is the headquarters of Monsanto, and one in Pennsylvania. So these are, and the non-Hodgkin's lymphoma is going to really, eat them up. I think that glyphosate is headed for termination of glyphosate usage worldwide. I hope so. Europe is fussing with possibly banning it. Yeah. And there's a lot of pressure on them now from activists to get it banned. And there's various countries in Europe that are thinking, oh my, maybe this is too tough. And of course, also Bayer has decided not to put Roundup not to put glyphosate in the Roundup, that product that they sell to the US personal market, not the agriculture. They're still happy to put the glyphosate on the corn, right, and the soy and the wheat and the garbanzo beans, but not on the dandelions in the yard. So they're,
Wait, but what do they use? What's the alternative? What do they do? Did they?
Well, that is the big problem, isn't it? How do you get rid of the weeds without having to hire a whole bunch of people to pull them by hand, which is really the best way to get rid of them, but also very costly.
There you go.
Very costly.
So here's the thing. The best way to remove weeds is by hand, because you're outside under the sun, getting exercise. We know that. We know monocropping is against what Mother Nature likes. There, in nowhere on this planet do you see monocropping. You don't. In the grasslands, that's monocropping, because grass is the perfect thing to grow on the prairies. But it's related to how deep the soil is and the microbes in the soil. And we know glyphosate kills that. Okay. So what I want to actually do is now I want to bring in, so we know glyphosate is bad. We can't really avoid it. It's kind of everywhere. You can't smell it. I have a hard time because, if I order in food, I know all these things, the sauces, the soy sauce, I don't eat any of that stuff. And I just end up cooking at home because it's too hard. I get really crazy about it. So I want to go into deuterium. And just for anyone who doesn't know what deuterium is, it's a stable hydrogen isotope. That means it's a different version of hydrogen. And where we're going to find this mostly is in water or in biologic molecules where there's a lot of hydrogen. Hydrogen is the most abundant element in the universe. It's also the simplest. It has one electron and one proton. And deuterium is a different version. It has one electron, one proton, and one neutron. There's another version of hydrogen called tritium. It's radioactive, and we don't have to talk about it right now. But in this universe, in this world, deuterium and hydrogen are the two forms that we deal with, and they're mostly incorporated into water, three forms of water: H2O, HDO, and D2O. Those are our options. When you're drinking water, that's what you're going to have. Heavy water is D2O. It's one oxygen and two deuterium atoms, and that's what they use to make nuclear weapons and all that stuff. So deuterium exists in our body on this planet. And I wanted to ask you, there's again, this is not we're going to go into some of the details, but it's interesting that there's more deuterium on the coastline as the water vapor goes higher. The water with the heavy hydrogen drops first. So we're going to find low areas of deuterium, high on mountaintops, further inland, and in glaciers for sure. Glacier water. So, and why don't you tell us about some of the features of deuterium that you thought were kind of, what to the average person, you're going to explain deuterium. Where is it, and where do we find it on this planet, and where can we expect it, and that type of stuff? Just to sort of a more like take a step back, and to the average person who's maybe not so into the kinetic isotope effect, but go.
Yeah, well, deuterium is everywhere. It's a natural atom, and it's part of, it's a kind of water, really. D2O is water. Actually, D2O looks a whole lot like water, regular water, and apparently it tastes a whole lot like regular water. But if you drink it, it will kill you. That's how different it is. So it would be a great way to poison somebody, to give them a glass of D2O. It's amazing how toxic it is. Incredibly toxic. Now, normally you don't have anywhere near that much deuterium in any kind of water naturally. It's going to be 155 parts per million is the standard amount in seawater. And most waters are somewhere around that. And in fact, there was a study done in the United States looking at the individual states, the 50 states, and they could get information on the average amount of deuterium in the drinking water in that state. And of course, the southern ones had more because they're further south. North is an advantage versus south, because as the raindrops come down, the water that evaporates is going to have less deuterium. It's a natural feature of deuterium that it stays with the liquid form. So when it's gas, it has less. And the same thing is true with the glaciers. The glaciers are ice, and they hold on to the deuterium. When the water melts off the glacier, the hydrogen leaves more readily than the ice, which is really important. So the glacier water can be down to like 90 parts per million, which is quite a difference. And they market glacier water on the web. There's a lot of different waters that they market, and there's a lot of confusion about exactly what is the health benefit of these waters. I know when I started hearing about all these different waters, hydrogen water is one that I find very interesting. And light water they sell, which is deuterium depleted. Oh, there you go.
Yes.
Deuterium depleted water, and light water can have extremely low. They sell it on the market, and I drink it. Actually, this has it in here right
Yes.
now. So
Yes.
it's 10 parts per million. So very low amounts of deuterium. You don't drink it straight up. You're buying it at. It's sort of interesting because you buy something concentrated, and then you dilute it with water. This is the opposite.
Well, I want to actually interrupt you there, because I've been drinking light water for a while. I know Robert Slovak, and it's his company, but I did a podcast with Victor Sagalowski, who's also, I think, a partner in that.
Yeah.
And we talked about a couple of things that are interesting, because I've been thinking about this for a long time. So I haven't diluted mine, and we talked about that. And he said that he was looking at, I can't remember if it was a case study he did where somebody had either esophageal cancer or something that was healed by drinking it straight. So we've been talking about just drinking it straight and what are the actual effects of that, and how quickly your body would assimilate it, but also that the water that you drink is less important than the food that you eat, because the food is broken down inside the mitochondria, and the water that comes out of that is what your body really wants to work.
Yeah, that's where you can get the really severely deuterium depleted water in the mitochondria as a consequence of the biology. And that is so fascinating. Yeah, and of course, it's the mitochondria that get killed by deuterium. That's the important thing too.
And there you go.
They hate deuterium.
Take us to the next level of this. So deuterium is a natural part of our environment. It's in our water. It's in our food. And then there are varying degrees of deuterium. Some foods are very high, like sugars and carbohydrates, and some are very low, like grass-fed organic fats. And glacier water is 90. The lowest food I'm thinking about is either, I think it's coconut oil or beef tallow, and they're both like maybe 110 parts per million.
Right. Yeah. Right? So you're coming down from 155 to 110, which doesn't sound like much, but I think it still is helpful. It's a big deal. All the fats, generally fats are low in deuterium if they're natural fats. And butter is a great one. Actually, I love butter and I use a lot of it. Okay. That's very like going to be 110. And also beef tallow, as you mentioned, also pork fat. Bacon, for example, would be a low deuterium food.
No. What about the GMO oils? What about soybean oil? Do you think that has a higher deuterium content? Because we talked about whether these oils are harboring glyphosate, and the difference is that an oil is a fat, and glyphosate is hydrophilic. And I think that Dr. Samsel had a theory about that, because when I think about GMO products, I'm thinking, well, first of all, corn is going to be higher in deuterium than olive oil or coconut oil because of how it's made, the photosynthesis. And then is deuterium coming along with that when you have something fried in those oils? So what do you think about that?
Yeah, I don't know. In fact, it's frustrating that there aren't very few measures that I can find, and maybe you can find more, but I'm having a hard time finding any data. There's very little data on how much deuterium is in different foods. And I think Laszlo Boros is one of the big leaders in doing that. It's not easy to test the foods for their levels of deuterium, unfortunately. So we don't know much, but certainly the fats are low. Cottage cheese, I believe, is somewhat lower than, say, sugars. And then the question of which fats is the interesting one. The animal-based fats clearly are. Breast milk is very low in deuterium. So breast milk is a source of low deuterium nutrient, and probably regular cow's milk is too, especially if it's organic. And I think the issue with the glyphosate is that glyphosate totally messes up the system's ability to handle the deuterium properly. Right. And so I predict that glyphosate-exposed crops and glyphosate-exposed animals are going to have higher deuterium in their nutritional products than the ones that are not, than the organically grown ones.
Right. So what you're saying is that is bad enough. So the starchy vegetables, they're higher in deuterium. But what actually is really happening is this combination, this tag team, I call it the tag team of terror. This glyphosate is uniting with deuterium, and together They're really destroying our biology, so that maybe on its own, deuterium, our bodies can handle it, but now we've broken the systems that deal with and remove deuterium, which is going to take us into sort of more details of actually how this works. And I really want to get down to the biochemistry, not because I'm really into it. I know the average person isn't, but I
wish they were. I really am frustrated with people that just say, "No, my eyes glaze over. I don't want to hear it. This is too boring."
Well, but I want to keep it sort of relevant, because I'm trying to, all my stuff that you'll see on the internet is really all about molecular biology, and it really is. And I think that it's important for people to understand that they're going to see the powers that be are going to try and fool us. They're already, they come out with these reports of the healthiest food, saying that cereal is better than eggs or meat. And I speak to people all the time about this. And so the problem is that they'll say, oh, there's a study about this or a study about, my favorite is the study that came out about vitamin D, saying it just didn't help with bone density, but there was no mention of vitamin K2. And I even had a patient who came in and said, you're wrong about vitamin D. It doesn't work. It doesn't make your bones denser. And so all these studies and all these indoctrination materials and all this propaganda comes together, that we have to actually be able to say, like, you can show me a million studies that red meat is not healthy, and I will say, I don't care what you say. I know that red meat, especially if it's grass-fed organic, I know this is one of the healthiest foods on the planet. Two reasons. Reason number one, it's one of the lowest in deuterium. Reason number two, the nutritional density of what's in meat is incredible. It compares to nothing else. So when I say the healthiest food on the planet is red meat, or the meat of ruminants, I believe that because these are the animals that really filter their food with their gut microbiome, and the meat that they put on, the cheese they produce is low in deuterium. So let's talk about enzymes, and let's talk about how does glyphosate make deuterium more toxic. And we're obviously going to talk about something really important, is the mitochondria. And the mitochondria is the powerhouse of the cell. We know that, but well, how can we tie this together, sort of without going too crazy?
You're afraid I'm going to go deep into the chemistry. It's really,
okay.
really fascinating. And I would like to start with the hydrogen water.
Let's do it.
Hydrogen water. And of course, you mentioned the gut of the cows, and it's so fancy, the ruminants, the fancy gut. They got a lot of gut microbes doing a lot of things. And one of the things they're doing is making hydrogen gas. And our microbes do too, but I think the ones in the cow do it better. They make hydrogen gas, and this is extremely important. And that's why hydrogen water is good for you. The same reason. It's the hydrogen gas. And when I first heard about hydrogen water, I was at a Weston Price meeting and someone gave a talk. I'm like, this is hocus pocus. How could it possibly be useful?
Right.
I really didn't have any clue how it could possibly be useful. Now I would say it's really outstanding. And of course, Mercola sells it. He promotes it, hydrogen water. And I sell
it on my website, because
there you go.
and there's hydrogen right there. And again, because when I got into the hydrogen stuff, I listened to Tyler LeBaron, who's very knowledgeable. And my question right away was, is this hydrogen deuterium depleted?
Exactly.
That's what I really wanted to know, because
that's what I wanted to know too, because it made so much sense to me. It occurred to me before I knew. I thought it must be, because when you think about the water coming off the glacier, less deuterium, right? Yep. And then I believe the gel that's formed all around the blood vessels is trapping deuterium. Jerry Pollack talks a lot about the gel being a battery and pushing out the protons. The protons are pushed out of the gel.
So I want to stop you there. For anyone who doesn't know, right now we're talking about something called easy water or exclusion zone water. Gerald Pollack is the one who wrote a book called The Fourth Phase of Water. I have interviewed him. His podcast is on my
There you go. You got all the good guys.
I got all the good guys, because I know, and I learned this right away from you, because again, we go back to that I'll never forget the way you had it drawn. You showed a blood vessel with a negative charge, and you showed a red blood cell with a negative charge, because you tied together why earthing isn't total baloney.
Yes, exactly. So when the negative charge.
Yeah, step on conductive ground, and you fill your body with electrons, and your red blood cells get repelled from the walls of the arteries if you have a healthy glycocalyx, which is going to be proteoglycans and sulfur is really going to hold water to it, easy water.
Sulfate,
Sulfate.
Sulfate is really important. It's sprinkled throughout the glycocalyx, and you can have less, you can have more. So there's all kinds of places where you can stick a sulfate, and your body does that. But if the sulfate supply system is broken, it puts in less. And when it puts in less, the power of the battery is reduced and the gel is less well formed. And the red blood cells are easier to get stuck. I mean, a lot of things go wrong. And the enos that you mentioned, I don't know if it's going to be part of the podcast yet, but you mentioned enos, which I've written about. Really fascinating enzyme, enos, endothelial nitric oxide synthase, produced by the endothelial cells lining the blood vessels. And I believe enos is a chameleon. It can produce sulfate and it can produce nitrate. It produces nitric oxide, that's well known, which gets oxidized to nitrate. I think it also produces sulfur dioxide, which gets oxidized to sulfate. And it titrates. It actually, the endothelial cells are watching the blood go by, and there's an electromagnetic signal that emerges from those migrating red blood cells. And that signal will be strong if the cells are moving quickly, and it'll be low if the cells are moving slowly. And so the endothelial cells in the wall have a way to tell how fast is the blood moving. And if it's moving too fast, I need to slow it down. If it's moving too slow, I need to speed it up. So you're looking for the viscosity of the blood, trying to keep it in that happy place. So you don't gel the water, you don't bleed out, very important. And it can go back and forth between sulfate and nitrate, because sulfate gels water and nitrate makes it liquid. And so the body's constantly juggling between those two, and it does it with the same enzyme, which is really amazing. When it's attached to the membrane, it makes sulfate. And when it's free from the membrane, it makes nitrate. And actually gathers near the cytoskeleton and makes the nitrate. So it's really, really cool.
Right. You may not remember, we did a podcast on that where we really went into the weeds on that. So if anyone wants to go really hear about this, the sulfate story is incredible. You're going to see one of my videos coming up for one of my foundational 4 characters, sulfur. And in the song, the words are deactivate, transport, lubricate, elemental electron aggregate. These are the things sulfur does, or sulfate does.
Yeah, really cool. I love your cartoon characters and their skits. It's so beautiful.
So when we're talking about this easy water, then we want it, because I interrupted you when we were talking about deuterium. So that this easy water, this gel, is actually potentially trapping deuterium.
It's sending out protons, and those protons are going to be low in deuterium. That's what I think. And there isn't proof, but it makes total sense when you look at the ice and the water. You look at the liquid drop coming down, the water evaporates, has less deuterium. It's always from the denser phase to the less dense phase. You've got the solid, you've got the gel, you've got the liquid, you've got the gas. At every step of the way, there's less deuterium. And when you get to the gas, it's tiny. And that is what's so amazing. So I thought it must be that hydrogen gas that's made by those microbes is low in deuterium. And of course, I started looking for a paper that would tell me that was true. Right? And I'm always looking for proof that this is true. And I found a paper from 1961, and it was beautiful. It showed that the microbes produce hydrogen gas by pulling it off of the organic molecules. It pulls the hydrogen off of molecules, pulls it off of carbon, puts 2 H's together, makes H2 gas, and that gas has 80% of the deuterium is gone, 80%. That is extraordinary, down to 20 parts per million. And that's why hydrogen water is so useful, because the hydrogen gas has much, much less deuterium than the hydrogen that's stuck to the molecules. What's left behind is going to be enriched in deuterium because of that. And then the really cool thing is that those hydrogen gas molecules are not just thrown away. It doesn't want it to just escape into the air. It wants to grab it back in and put it back on the carbons to produce organic matter that's reduced in hydrogen. So there's a titration process that goes on by making the hydrogen gas. And the gut microbes know this. They know they have to do that and bring it back into organic matter. It's so cool. Right. I love that.
So what you really discovered is that there's these the microbes and the enzymes themselves are, and this is what I think is the most fascinating thing, is that they're constantly positioning themselves to make sure that the hydrogen they're grabbing is hydrogen, not deuterium.
Exactly.
Exclusively, except in some conditions, which I found fascinating. I think it's petroleum. So
right, it's amazing, isn't it? It wants the opposite. It wants deuterium, not hydrogen.
It wants deuterium, and what's fascinating is that I thought that you never want more deuterium; you want to get rid of it. And then I actually started talking with Doris Lowe. I don't know if you know, and so it's a whole. And I started saying, well, what would be good about deuterium? Well, if it's incorporated into collagen,
exactly,
it would strengthen collagen, and that kind of blew my mind. So
that is right, and that is what's so cool. That's where I see this dichotomy between the collagen and the gelled water wants to trap deuterium. And then the water that gets into the mitochondria wants to throw it away.
Got it. So basically are the structural elements of our body that are made of collagen, they're stiffer, they're stronger. Your fascia, that's going to have a higher degree of deuterium, because when you add that extra neutron, you increase bond strength. So you actually get a
Yes,
that's stronger.
stronger bonds, and there's a lot of proline in collagen. And in fact, remember, and there's so many things that I learned way back when, and then once I know deuterium, I look at them again. I say, oh yes, of course, that's why. Because I was noticing that I found it fascinating, and of course, I had no clue why. I'm always looking for interesting things in biology and asking why. Why did it do it this way, right?
Yeah.
So I realized early on, long before I knew about deuterium, that the structural proteins have lots of proline in them. Categorically, they have lots of proline. I mean, collagen probably has more proline than glycine. It's got a huge amount of glycine and a huge amount of proline. That's almost everything in collagen is either glycine or proline or hydroxyproline. There's just lots and lots of that in collagen. And so the structural proteins have a lot of proline. And then the enzymes have low proline. They don't have much proline. And I was like, well, why is that? What is it about proline that makes the body want to put it into the structural proteins? I thought it's got to be a good reason for that, but I didn't have any clue what it was. Now I know. It's because proline can trap deuterium, and you want deuterium to be in the structural proteins because they're not enzymes. They're not trying to make a product that's low in deuterium. They're just trying to trap the deuterium to keep it away from the rest of the system so that the fluid water will be low. So when you think of the blood, all the vessels surrounded by this gelled water with all this collagen and all this deuterium trapped in the collagen, then trapped in the gelled water as well. The gelled water has higher deuterium than the fluid water. And they're sucking all the deuterium out of the fluid blood. So now the blood has low deuterium as a consequence of all of that.
Wow, fascinating. All right, well, so I know this may be confusing, but I love this topic. So I want to just talk about the kinetic isotope effect and deuterium. And I want to talk about really, so the most fascinating thing for me is to think about how our bodies evolved and how all biology evolved, and it all really evolved to avoid deuterium.
Yeah, I think it's an absolute central feature of biology.
Yeah, and how crazy that as these enzymes evolved, they were preferentially looking for that H instead of that D. It's kind of crazy. So, I also want to sort of put this question to you. There's the different theories of aging, but no one's really putting out the deuterium theory of aging, which I think is more relevant than the free radical theory of aging, because deuterium can increase your free radical
deuterium, the deuterium dysbiosis creates free radicals. And that's how the mitochondria get wrecked, because when you have too much deuterium in the mitochondria, the ATPase pumps get gummed up. It's like sugar in the gas tank. They get gummed up with deuterium. They don't work well. They spew out reactive oxygen, and they don't make as much ATP. They waste the energy making reactive oxygen, losing the ability to make the ATP.
So this is our opportunity to talk a little more detailed on how that all happens. This is where glyphosate and deuterium really merge. Is that when we look at the part that I want to be is don't find that interesting, or we already have talked about, is that how the mitochondria produces energy is that it creates a proton motive gradient. The protons want to come into the mitochondria, and as they come in, they spin an actual rotor. You're going to see in my work, you're going to see my logo for Deuterium Records is actually a cross section of ATP synthase, and with the actual chambers that ADP, adenosine diphosphate, and the inorganic phosphate are put together. One of the most incredible things I did was spend a lot of time taking apart ATP synthase, because this is how all multicellular life evolved on this planet. Mother Nature created this engine that powers all life, that's universal. And so this rotor is spinning like a hydroelectric dam. And when it's run by regular hydrogen, but when a deuteron or a deuterium atom goes into it, it breaks the nanomotor, or it stutters and slows it. And that's what the mitochondrial damage is. But this is normal to a certain degree, because we can't protect the entire mitochondria always from deuterium. But
you're never going to get rid of all of it. Yeah.
Right. So then let's talk about, how does glyphosate go in there and damage the mitochondria?
It's two things. I would say two principal things. One is the inability to make enough sulfate, so you don't trap as much deuterium in the extracellular matrix. You've got more deuterium in the blood. You've got generally more deuterium in the fluid water, because they're not trapping enough in the gel. That's number one.
So number one is a sulfate deficiency caused by glyphosate, because glyphosate is a chelator. It's going to take those certain minerals and take them out of action. So
It's
that's
not just the mineral sulfur, it's also the enzymes that deal with the sulfur. And in particular, at least I would say there's several enzymes that are involved with sulfur, making sulfate, attaching sulfate, all these things that are negatively affected by glyphosate. And this is something I talk about in my book, and it has to do with glyphosate's demonic, insidious ability to substitute for glycine during protein synthesis. This is crucial. Right. And I believe that I talk about in my book a glyphosate susceptibility motif. And that is a protein that has a glycine residue that's very important to that protein. You can see from the literature that if you swap out glycine, put in alanine, the protein's dead in the water. I mean, like even a minor change messes it up. It has to have glycine there. And there are many, many enzymes, important enzymes that are involved with managing deuterium that have at least one, sometimes 3 or even 4 glycines at the place where they bind phosphate, That's the critical thing, because glyphosate has a methylphosphonate unit attached to its nitrogen that can fit in that spot that's supposed to, where the substrate phosphate is supposed to go. There's room for glyphosate to put that there. And that makes it easier for it to substitute.
So that's good. I want to slow you down. Just so what you're saying is, we know that in our bodies, we're made of 20 amino acids. The smallest and simplest of all those amino acids is called glycine. It's very important because it's required in collagen. Because it's so small, it's required for tight turns, and it's in a lot of enzymes. Now, the biggest issue is why glyphosate is so dangerous is that this is an imposter or an analog of regular glycine. So when you're talking about an enzyme, we know that all enzymes they're amino acids that are stuck together one after another, like
in a line,
string, and then they fold to create their ideal form. If you take a glycine out of an enzyme and you replace it with a glyphosate, the body will do this because they're so similar, but now you have this piece that looks like glycine, but it has an extra piece sticking off, and that extra piece starts to interfere with folding. And not only that, but it interferes with the active site. Enzymes have an active site or a business end where they're going to do their reactions.
And so you need that room for the phosphate. When you're going to bind a molecule that has phosphate, typically you're going to want to stick to that phosphate, and that phosphate needs a place to go. Well, glyphosate says, oh, that's a perfect place to put my methylphosphonate because it looks a whole lot like phosphate. It fits in the same place. It has the same negative charge. So it's like, great, I can put it there. I can go into this enzyme. In the way that it folds, there's room for my extra piece. And that's really critical. So glyphosate won't substitute for glycine everywhere. If it's crowded with a bunch of big amino acids next door, it's not going to fit, But you have you have sequences like GAG. That's a sequence that binds a phosphate of ATP, and it has glycine and alanine, which are the 2 smallest amino acids, a string of 6 of those that are all tiny amino acids. There's plenty of room for glyphosate's methylphosphonate.
Right. So what you're saying is certain enzymes specifically are more susceptible to substitution of glycine for glyphosate. And I want to bring up, so most of my worldview is vitamin D based. And I initially found your work through your discussion on vitamin D sulfate. And just so anyone knows, I see all this stuff on the internet, on Instagram, saying, you don't know, there's vitamin D sulfate, like this is some magical thing. I'm like, okay, vitamin D is hydrophobic. It has to be bound to something to get around.
Yes.
And vitamin D sulfate is potentially how your body really wants to transport this stuff.
Perfect.
There you go. So one of the things.
But then people say, oh, well, vitamin D sulfate's not interesting because it's inactive. They say it doesn't do anything. So you don't want vitamin D sulfate, but they don't understand it. It's a transport system for both the vitamin D and the sulfate. Neither one can move around without the other.
Well, there you go. And so why is your story on sulfate is so critical? Because a lot of the hormones that we want to move around, we don't want them where they're made, and where they need to act is different. So because sulfate will deactivate,
it's a perfect solution. And both deactivate and transport. It's so beautiful.
Yep.
And that's true for cholesterol and for all the neurotransmitters and the hormones, the sex hormones. I mean, it's amazing. All these lipophilic molecules that don't like to hang out in water.
There you go.
You stick a sulfate on, you send them out the door, you know, and that
Okay.
inactivates them at the same time. You don't want them to be active when they're floating around in the blood. You want them to be inert, you know?
Right, right, exactly. And so the problem is that there are specific enzymes that will attach whatever, vitamin D, cholesterol, whatever, to sulfate, and those enzymes are being damaged by glyphosate.
That's right. And in fact, the enzyme that produces the universal sulfate donor is called PAPS synthase. And that enzyme has a critical sequence in it. And I think it's even GAG, but it's something like that. It's got 3 or 4 glycines at the place where it binds phosphate. It combines the sulfate with an ATP molecule, makes some interesting changes, puts out this PAPS, phosphoadenosyl phosphosulfate. PAPS is a universal sulfate donor. And that enzyme, I believe that enzyme is being suppressed by glyphosate. In fact, it's shown to be suppressed in E. coli in a study. And I can see why, because it can put itself there with that glycine. There's multiple glycines at that site that bind ATP. And many, many, many enzymes bind ATP at a site where they have at least 1 off to 3 highly conserved glycines. Tremendous opportunity for glyphosate's mischief. And I think it's happening to all of them. And that is really crucial. So the sulfate's not getting activated by that enzyme. And once it can't be activated, it can't be hooked onto anything.
Got it.
That's a major block.
All right. And so basically this little, monster glyphosate is interfering with our ability to sulfate everything, and as a result, interfering with our ability to have a healthy glycocalyx, to have a lot of easy water where it's supposed to be. So this is really, and so it's letting more deuterium out into everything.
That's exactly right. Yes.
Okay.
I mean, you can see how that would generalize into a huge problem.
Yep. And now as we go to the mitochondria, again, we know that we don't want those deuterium atoms coming into that nanomotor because they're going to break it. And then how else does glyphosate affect the loading of the electron transport chain? How can you?
Yeah, so that's the other piece of the puzzle, and that one's also very beautiful and of course terrifying at the same time. And that's the dehydrogenases, a whole class of enzymes, dehydrogenases. There's lots of them. Many of them are in the mitochondria. And the ones in the mitochondria actually they extract hydrogen from some organic molecule and they attach it to NAD and they make NADH. And there's several of them in the mitochondria. Then there's one that takes NADH. That's an NADH dehydrogenase. It's got its code in the mitochondrial DNA. Wow. Very important enzyme in the mitochondria that takes the H off of the NADH and puts it into the mitochondrial intermembrane space. So it's basically all the organic molecules have these hydrogens. They're extracted by these enzymes, these dehydrogenases. They're stuck onto NADH in most cases. Another one is FADH2. Those are very similar. Both of them pick up those beautiful hydrogens from those molecules and provide them through NADH dehydrogenase into the intermembrane space, so to supply the ATPase pumps with low deuterium hydrogen. And those in E. coli, there's like a dozen dehydrogenases that are suppressed by glyphosate in E. coli. Right. That's a gut microbe.
So I know that even though you maybe not be participating in the biohacking world, because I saw one of your last interview, they're asking you biohacking questions, and you're like, I don't follow that stuff. But a very big thing is NAD supplements. Everyone's getting NAD. So I'm thinking that the reason they're having to bolster their NAD is because basically it's not functioning the same way. It's getting killed by glyphosate.
And it's also being lacking. NAD itself, even without the H, is deficient because glyphosate disrupts this critical enzyme in the shikimate pathway that produces tryptophan, and tryptophan is a precursor to NAD. So,
wow. So
it's actually the supply of NAD is reduced by glyphosate, and the ability to stick the H onto the NAD, and the ability to take the H off of NAD. All of those are disrupted by glyphosate.
Wow. And so just to tie this together, so this new toxin that's an imposter for one of our natural components of our bodies is at every step of the way interfering with our enzymes' ability to remove deuterium.
Yeah, it's just a mess. Both trapped deuterium and removed deuterium, both of them are problematic with glyphosate. It is a complete train wreck for deuterium.
Fascinating. Okay.
And
So
I want to go back to the hydrogen
yeah,
gas, because
please.
I want to
Okay,
cover
sorry, we
that
got to
piece of the
interrupt you there.
puzzle as well.
Yeah, please go
Yeah, we
on.
got that hydrogen gas now. It's in the gas. What happens to the hydrogen gas next? Because it doesn't just get thrown away. It would just escape into the universe if you don't trap it, right?
No, and just to let people know, so this hydrogen is so small that when the gas is made, it can go around your body. It's not bound, so it can.
Right. It can move through tissues. I mean, it's so tiny, it can just escape into the universe very easily. So you need to bring it back quickly. You got this great hydrogen gas, really low deuterium, we need to grab it.
Yep.
And you have enzymes that can do that. Gut microbes, again, they take the hydrogen gas, they take carbon dioxide, they put those two together, they release oxygen, they get rid of the oxygen, I guess they maybe make water, and then they grab the hydrogen and they make methane, MH4 methane. And you've probably heard about methane being a greenhouse gas. The cows, we need to kill the cows because they're producing so much methane, right?
Yeah. Yeah, terrible.
And they're producing all that methane because glyphosate is disrupting the enzymes that capture the methane and keep it into the body. So the methane gas just escapes, and that's why we have so much more methane from the cows, because they're being poisoned by glyphosate.
Wow. Okay. So first of all, I don't believe any of the stuff that cows are making too much methane that's causing global warming. But aside from that, so basically the cows could be releasing more when they're eating GMO corn, grain, and soy rather than grass.
Exactly.
They're actually releasing more methane because the enzymes that we're going to try and keep it are
yeah. So you have the hydrogen gas, you don't want it to escape, so you quickly turn it into methane gas. You don't want it to escape because that's still a gas. You're like, oh my god, it's still got a gas, I got to do something. They make methanol. Methanol is the smallest alcohol. Ethanol, of course, is the one everybody loves to drink, right?
Right.
2 carbons. Methanol's just got the 1 carbon. It's the CH3OH. So it's taken one of the hydrogens, turned it into a hydroxy, and that keeps it from being a gas.
Oh, got it. Okay.
That's how you capture it. You've got to go boom, boom, boom. You got the hydrogen gas. We've got to make methane. Oh, we got to make methanol, and then you can go on and make formaldehyde and formate by keeping on changing H's into OHs. And when you do that, you stack those H's onto NAD. So you make each one of those dehydrogenases loads up an NAD molecule with a golden H coming straight from hydrogen gas, extremely low in deuterium.
All right, and again, to anybody, this hydrogen is being fed into the mitochondria as a fuel source, and the reason that this all technically should work is because it's preferentially selecting at every turn a hydrogen over a deuterium, unless we
right. Each one of those dehydrogenases knows how to select hydrogen over deuterium, but on top of that, they're working with molecules that already have low deuterium because it came from the hydrogen gas.
Got it.
So it's really golden H on those NADs that came from this pathway, traced back to the hydrogen gas.
Right. All right. So I want to just want to tie some other stuff together. So when we look at vitamin D, because again, it's a big issue with me. So there's an enzyme, the CYP enzyme, CYP if it's number 27, the enzyme that activates vitamin D. Because I see all these kids around here who I know are vitamin D deficient because I could see the signs the parents say, oh, they grind their teeth. I know the kids are low in vitamin D. Yet at the same time, they're spending time outside or they're getting supplements. So my thought process is they may have a very high level of vitamin D in their body, but they can't even put it into the storage form. It's just vitamin D. It can't even be the 25-OHC, never mind the active form. So right. Glyphosate itself, because I believe that the enzyme, these enzymes that activate vitamin D, they have susceptibility to glyphosate.
They're another class. Cytochrome P450 enzymes, really important in the liver, also in the kidney. Vitamin D is really fascinating because it goes to the liver and it gets 1 hydroxy added, and then it goes to the kidneys and gets another hydroxy added, and now it's ready to be active. It has to go through those 2 steps. I believe it's actually it's a test. Vitamin D is a probe, and it goes to the liver. It says, oh, great, the CYP enzymes are working. Let me go check out the CYP enzymes in the kidneys. Oh, good, they're working too. Now we can go ahead and do all this great stuff because things are healthy. But if those enzymes are not working, the vitamin D just gets stuck and never gets activated because it can't get through those pathways because glyphosate's disrupting those CYP enzymes.
It would be fascinating to see if anyone would be testing, especially kids who are, because I tell all my patients, I want them to supplement D3 and K2 and magnesium very profoundly, because I've been trying to sound the alarm for the pediatric jaw growth and development. To anyone who's watching this, if you haven't seen my work with what I call long face syndrome, as a dentist, this is what I'm seeing in all my kids. is that they all have a tongue thrust swallow. They have very narrow palates. Can't breathe through their nose. They have a lack of calcium in their jaw. And so vitamin D deficiency with vitamin K2, this is at the root cause of a lack of room for all of our teeth and decreased size of airways, because pediatric sleep apnea is massive now. And they're putting
Interesting. I didn't know that. That's
Yeah.
very interesting.
Yep.
Wow.
And so because I know that the vitamin D deficiency to me connects so many different things, because vitamin D also regulates the gut microbiome. So if you have vitamin D and you can't activate it, you know, your gut microbiome, I don't know if it can get to, probably can get the vitamin, hopefully they could get the vitamin D as well, but they have to activate it as well. So when kids are eating a lot of processed foods, and that's pretty much all they're eating, these kids are all turning up vitamin D deficient. And nobody's testing kids' vitamin D because it requires a blood draw, and nobody wants to draw blood on kids. I worked for 2 or 3 years trying to get somebody to do a urine or a salivary test for vitamin D, to show these patients, these babies are being born with tragically low levels. So that's one of my pet peeves. But where it ties in with glyphosate is that glyphosate is destroying the enzymes, the COIP, cytochrome P450 enzymes. And I have cartoons for those. If you download my book,
Excellent.
you're going to see them. And I have them as they're shiny and bright, and they defend your body, because these are the enzymes that detoxify chemicals in your liver. Right?
Yeah.
Okay.
Right. Absolutely.
So having said that, it makes sense that vitamin D deficiency, when people say I'm in the sun all the time, and then they're eating all these foods that are destroying their ability. So when they go for a test, they don't have that 25 OHD level. And they're like, I'm taking supplements and I'm in the sun. So that's the first thing. The second thing is I want to talk about B vitamins. And the reason I want to talk about that is because in my thought process from the work that I've done, vitamin D regulates the gut microbiome and allows the species of the gut to make all of the B vitamins. And this is work that I've heard through Stasha Gomanak. I've done a lot of podcasts with her. And I found her because she was saying sleep apnea root cause is low vitamin D. And that's how I got into vitamin D in the first place. And what she discusses is basically a B5 pantothenic acid deficiency, which causes a deficiency of acetylcholine. And acetylcholine
that's interesting. This is a path I haven't followed. I've studied
so
a lot of the B vitamins.
Yes,
I've missed B5. It's not one that's on my radar screen.
Well, it was interesting.
That's really interesting. That's linked to acetylcholine.
Yes,
that is amazing.
And what her finding was that thinking about this, when you take vitamin B5, and you should take the B vitamins as a complex, but it's going to increase your ability to produce acetylcholine, and acetylcholine calmness and mental clarity during the day and allows you to sleep at night. It's the chemical that's going to let you go from fight or flight into rest and digest. It's the primary neurotransmitter of the rest and digest autonomic nervous system. And
I need to tell you about acetylcholine because it goes directly to deuterium in a really big way.
Well, let's hear
in a really big way. Story. So choline, do you know what the molecular formula is for choline?
Not off the top of my head.
Well, it has a nitrogen, and that nitrogen has 3 methyl groups attached to it. 3. And when you trace where those methyl groups came from by the chemistry of how that is made, they came from methane gas. They came from methane gas produced in the gut. So in other words, those methyls on the choline are gold. They're pure gold because every one of them's got 3 hydrogens that are low in deuterium. Yeah. And then on top of that, there's the acetyl, right? Acetylcholine. Acetyl is a methyl group with a COOH attached to it. And acetate also comes, there's gut microbes that make acetate. These acetogens, right? They make acetate from methane or from hydrogen gas. They go from hydrogen gas and carbon dioxide to acetate. And the acetate has a CH3, that's the methyl group, and then a COOH, that's the carbon dioxide. It basically attaches carbon dioxide to a methyl group. So a little different from methane, just because it has that COOH attached to it, but it's got 3 hydrogens there that are gold, pure gold. So you've got the acetyl group having a methyl. So you've got 5 methyls. You've got 5 golden methyls on acetylcholine. That's why it's so valuable to the body.
Fascinating. Okay.
Very low deuterium.
So what I'm seeing, and these are kind of really interesting things. So with something called bruxism, clenching and grinding your teeth. So there's an epidemic of bruxism. I'm a dentist and I make grinding guards for all my patients. But what I've found and what's really fascinating is with young kids. So when a 6-year-old or a 5-year-old, you can hear that grinding noise coming from their room, and nobody knows what's causing this. It's certainly
Wow.
not stress. And when you dose a child with vitamin D, they will stop grinding their teeth within 2 to 3 days sometimes.
Wow.
But
That's
not only that,
fascinating.
so adults, if they have the right level of vitamin D, they will stop grinding. But not only that, the B vitamins themselves will decrease grinding. So if you're listening to this and you clench and grind your teeth, and you take a B complex, and within half an hour you actually stop bruxing, I think that's really profound.
That's incredible.
Yeah.
Yeah.
So
Do you
Dr.
have a theory for what's causing them to grind their teeth? What are they
Yeah,
accomplishing?
acetylcholine deficiency, B
Yeah,
vitamin
but what
deficiency.
are they accomplishing by doing that?
It's a short circuit. It's a neurological short circuit. The part of
Wow.
the brain that controls the airway, this is the most, so I usually like to say this, that you can live for many days without food. You can live for quite a few days without water. You can't live for more than 3 or 4 minutes without air. So our
Mhm.
bodies are really paying attention to oxygen as the highest level of interest. So the part of the brain that regulates sleep and breathing is very, very fine-tuned for all this stuff. And it's supposed to be sending a very clear signal. But if the neurotransmitters aren't being made at an adequate amount, because I see it as a two-prong issue. I see it as a lack of the right B5 that has to get made into acetylcholine. But not only that, fascinating that choline acetyltransferase, the enzyme that makes acetylcholine, it's made in the soma of the neuron, and it's transported down to the synapse by dynin and kinesin, by motor
Interesting.
transport enzymes, and they run on ATP. So if you don't have enough ATP, if you're not producing enough ATP, these motor proteins that use one ATP molecule per step, you're gonna have a
That's
hard time
interesting.
transporting your choline acetyltransferase, the enzyme, because what your body does is it takes that, it makes it near the
In the
synapse. So
soma. Yeah.
yeah, sends it down there to be assembled. And if you look at the recycling of acetylcholine, you can see there'll definitely be, if there isn't enough ATP, you can't transport that enzyme down there. So my theories are, so when I don't take the right B vitamins, I don't sleep properly and I grind my teeth. And I have seen sleep apnea disappear with the right dosing of B vitamins.
Yeah, that's really interesting. It
So
looks like the B vitamins play a huge role in managing deuterium because the other ones, folate and cobalamin, they're involved with the methylation pathways. People probably know that because there's a lot of talk on the web about autism being connected to methylation pathway deficiencies. And the methylation pathway is taking those golden methyls that came out of methane and moving them around. The body keeps track of them. It actually is quite interesting because methionine is a universal methyl donor, just like you have the PAPS. PAPS is a universal sulfate donor. Methionine is a universal methyl donor, and methionine comes from methane. It's basically a methane thiol, S methane. It's a methane with an S attached at three hydrogens and an SH. Just like you have three hydrogens and OH, that's the alcohol, methanol. And methane thiol is alcohol, except you got an S instead of an O, if that makes sense. It's a sulfur. And that methane thiol gets attached to an organic molecule to make methionine. Again, the gut microbes make this. We can't do it. We depend on our microbes to make the methionine. And that enzyme is the enzyme that makes the methionine is suppressed by glyphosate in E. coli.
okay, well, I can assume it's probably in other places. Okay, so that was really, we really got into the weeds on that. So when it comes to, these are my theories. I've talked about them before, but so a B vitamin deficiency, in my opinion, is really at the core, at the heart of why we have a lot of mitochondrial dysfunction, because the B vitamins are instrumental in the electron transport chain, right? They're involved in the production of energy itself.
Yeah, they're so important. And in fact, I'd mentioned Zen Honeycutt doing the tests on the glyphosate on the foods from the fast food industry, like 20 different top fast food industry players, food samples from them. She did a big analysis, and not just glyphosate, but a lot of other things as well. She looked at B vitamins, and they were in the toilet. Cobalamin and folate were zero. They're undetectable in any of the samples, which is really, really shocking. And those are the ones that control the methylation pathway. I should go back and look at B5 because I don't pay attention to B5, but I'll bet it was low too, because pretty much across the board, the B vitamins were really low in these foods.
Yep.
So when you eat processed foods from fast food industry, you are getting a low B vitamin diet for sure.
All right, well, because in my, I like to simplify everything, as you know, with cartoons and that. And if you take so I have two D's make a B. So vitamin D, a D on top of a D makes the letter B. And vitamin D regulates the gut microbiome to produce B vitamins. And so, what I really believe is that we're seeing just a global epidemic of low B vitamins, and those low B vitamins result in low acetylcholine and the destruction of our sleep. And
That
then
makes sense.
all the other paths, and mitochondrial dysfunction. If you don't have B vitamins, you can't produce energy.
Yeah.
So, all right. Okay, so I'm sorry that we definitely got into the weeds. So now I want
so sorry.
It's okay. Well,
It's fascinating to me.
I want people, so because there's going to be, I've watched a lot of your podcasts. I've watched other podcasts. At a certain point in time, there's certain specific people out there in the science industry, in the biohacking industry. This conversation is great. For some people, it's going to be too much, but a lot of people are going to be saying things like, "Oh, sounds like baloney" or whatever. So the more detailed you are, I love the way you talk about these things, because to me, it's always a puzzle. Everything
Yeah,
has,
same here. I love the
yeah,
puzzle, and I always ask why. Whenever biology is doing something, there's a good reason
there's
for
a
it.
reason. All
You
right.
just don't know what that reason is, and sometimes it's really hard to figure out.
Definitely. So then I want to sort of just maybe finish up with your thoughts about what's the best way to live a deuterium depleted lifestyle? What do you think? What are the things that people can do to keep deuterium out of their life? Or I guess it's definitely a combination of the deuterium and glyphosate. We've talked a lot about how to keep glyphosate out of your life. It's pretty tough. But what are your thoughts on, what would you recommend to the average person when it comes to this stuff? What's your thought?
Yeah, I mean, I generally tell people to eat a very wholesome diet, organic, certified organic, whole foods, cook at home. Don't buy any processed foods, don't buy any fast foods. Try to minimize your glyphosate exposure, largely through diet. But of course, you don't want to live next to a farm where they're spraying glyphosate. That would be really bad.
Right.
And even a highway, because glyphosate is showing up in the cities because I think it's escaping from gas. You have these bioethanol and bio diesel fuel. These things are contaminated with glyphosate, I believe, and that's escaping into the air before combustion. And so you've got glyphosate in the city. So you don't want to live near a highway. You don't want to live near a farm.
Yep.
And you want to eat a wholesome diet that's whole foods, cooked from scratch. And then, of course, sunlight exposure is very, very important for health.
Well, so let's talk about why sunlight exposure is so important, aside from vitamin D. And because it's funny, because Laszlo Boros basically said, because I was like, really stuck on vitamin D and sunlight. And he said, oh, vitamin D will help against deuterium. And what was interesting is he said that the resonation kept the matrix water less viscous.
Oh my, that
Yeah.
sounds wrong to me.
Well, so this is when vitamin D, because you've broken the ring, the carbon ring, it's going to resonate differently. This was a long time ago, but I like that idea of like, why? Because I feel like as the vitamin D level goes down, the deuterium goes up, and I feel like they're tied together.
Yeah,
But
well, it could easily be the sulfate, because the vitamin D is actually pushing sulfate around, just like cholesterol is. The skin produces a lot of cholesterol sulfate in response to sunlight. It catalyzes the production of cholesterol sulfate and vitamin D sulfate in the skin. And both of them are escorting sulfate around the body. So vitamin D is, in a way, if natural vitamin D is a marker, it's a small amount compared to the cholesterol sulfate that the skin releases. The skin releases a lot more cholesterol sulfate than it does vitamin D sulfate. Vitamin D is small levels. It's a signaling molecule. But cholesterol sulfate is probably the workhorse. It's delivering all that sulfate across the body.
Right.
And the vitamin D sulfate is an indicator. It goes to the liver and it says, oh, good, we've got cholesterol sulfate because here I am. And I came from the same place the cholesterol sulfate came from. Just a tiny amount of me, but you got lots of cholesterol sulfate up there. I can reliably tell you that's the case. Of course, that's only if it was actually made in the skin. When you take gobs of vitamin D as a supplement, now you've got a false signal coming to the liver. Oh, it must be lots of cholesterol sulfate because there's all this vitamin D. And it's not true, because it didn't come from the right source. So that, I think, is a problem with supplements.
Right. Well, so there's definitely there's again the influencers are talking, and they're saying a lot about that. So I want to always stick up for vitamin D supplementation, because as I got into this, I wasn't thinking about going in the sun. I was thinking about what pill do I take, and I was able to raise my vitamin D level with supplements alone. And when I had my health improvement, I started to think, well, I should definitely get this naturally. And I don't take any vitamin D until we get into the time zone change, or
Hmm.
you know, I've got a job, so I don't get to go outside as much as I want. But we did a whole podcast, again, you won't remember this because it was quite a few years ago. We did a whole podcast on cholesterol sulfate because I'm fascinated by that. And you know, in my cartoons, I have cholesterol and sulfur. In the video that I sent you, sulfur, he's got a crush on cholesterol.
I know, that's so cute.
Yeah, and I call them the new power couple, cholesterol sulfate.
That's so beautiful.
Yeah.
And it's true. And both of them depend on the other to be transported. So when you don't have an ability to add the sulfate to cholesterol, which is because those enzymes are being messed up by glyphosate, now you've got a problem with transporting both cholesterol and sulfate. Both of them are in trouble.
All right. Now, is there anything else? Okay, so being in the sun also is going to build your easy water, which
Yeah.
is going to trap more deuterium. So just being in the sun is going to deplete deuterium.
Exactly, because it's going to increase the sulfate supply to the glycocalyx, which is going to trap the deuterium. So that's very straightforward.
Okay.
Right.
Then now
grounding is good too, right? Walking in on the beach in the water. I luckily live near the beach, you know.
Right. Well, so how will grounding affect deuterium?
Well, yeah, that's a good question. Grounding provides you with that negative charge,
Yeah.
which
So
will
technically,
help
yes.
the circulation.
Well, it would also help to build more easy water potentially. So
Probably so,
all right.
yeah.
Now, Jack Cruz has come out saying that a tan, the melanin, traps deuterium.
Oh my.
Yeah.
I don't know where he got that.
Well,
oh my, I don't know.
You know, I think it's fascinating just because also, you know, so basically your tan, if it traps deuterium and it exfoliates off, that makes sense to me as a way to deplete deuterium. So
Yeah,
I'm not sure.
yeah.
I guess, you know, melanin is such an interesting molecule because it absorbs everything. All light is absorbed. So maybe,
Yeah.
you know,
It's a protective. I mean, in fact, melanin, of course, protects you from the sun. That's a natural protection, not natural sunscreen, if you will, right? Melanin.
Yep.
And a lot of people have told me, you know, I get out in the sun, I never tan, I just get red, and then I just get white again, you know. And I'm okay, melanin deficiency, because that comes out of the shikimate pathway.
There you go. So to slow down for people, if you can't tan or you can't create a tan, you need to understand that glyphosate itself is affecting the bacteria in our guts that make the precursor to melanin. And
that's right.
You know,
that's right.
I've been calling it the shikimate pathway because it's more Japanese sounding.
That's probably the right pronunciation.
I
But people seem to call
it
shikimate. I wanted to call it shikimate too, because that seems right for.
Well, I call it.
Yeah, you honor its appropriate pronunciation rather than going with the crowd.
Well,
you know,
I have my seven shikimate enzymes. They're the 7 sisters, and I have them. They're drawn as enzymes, and they're very Japanese anime because I like that. You know, I really
that's
like
nice. Yeah.
Okay,
that's nice.
So I want to finish our discussion with something that's really going on. A lot of people are talking about it. It is related to COVID, but I want to steer away from COVID. I want to really talk. I'm not that interested in talking about it. I want to talk about heart disease. When I read your material on cholesterol sulfate, I knew right away that you were onto something, and that the idea that you eat butter and eggs and that cholesterol goes into your arteries and clogs it, that's nonsense. And I call it non-scientific extrapolation. Seeing butter and seeing what it looks like, and then seeing cholesterol in your arteries, like, oh, that butter got in there. That part of my language, that's bullshit. I don't believe that for a second. So then what is the root cause of cardiovascular disease? And so in my thought process, based on your work, what I've learned from you and a lot of other stuff, it's all about the glycocalyx. If your glycocalyx is denuded, if it's gone, it changes everything. And isn't that going to be a primary site? So I think that heart disease, and you can tell me what your thought process is, the vitamin D3, vitamin K2, magnesium, and sulfur deficiency. It's all that. And the primary initial damage is the destruction of the glycocalyx. So now
I agree with that.
Remove this protective coating from your arteries. And then we also have to think about vitamin K2 deficiency. Now you've got calcium building up on whatever cholesterol plaques that you have. And doctors are looking at this thinking, oh, we're going to do a CAC scan because you have calcium building up in your arteries. Well, why do you have that? Because you don't have vitamin K2, because your enzymes can't remove it from your blood. For vitamin D, there's a million different things that can affect, but primarily the renin-angiotensin system. There are studies out there that show that if your D level is low, that system gets broken down. But I'm hearing about ApoB, that everyone wants to think about these boats that are ferrying around cholesterol and vitamin K2 and all these fat soluble vitamins. They want to blame this. What are your what's your thought process about that? Because I listened to a whole podcast with Tucker Goodrich, and he would bet his life that it's this ApoB that is getting absorbed into the blood vessel that's causing these cardiovascular plaques. And I'm thinking, well, you didn't mention vitamin K2 and you didn't mention the glycocalyx. So how much can you really know about that?
Right. Yeah, it's all very complicated. And of course, none of us knows for sure what's going on. But I actually think that it's quite simple. The liver does produce high levels of LDL when you can't sulfate the cholesterol, because you have to hide it inside the LDL particle. Now, if it's not sulfated, it's not water soluble, you can't just ship it out. You got to package it up. So the liver has to make more LDL to deliver the cholesterol when it can't sulfate the cholesterol.
Okay.
And in fact, it's quite interesting because there's this, PCSK9 inhibitors. Have you heard of them? PCSK9, that's the big new thing in controlling your cholesterol. People who can't take a statin drug, they give them this PCSK9 inhibitor, which inhibits this molecule, this enzyme in the liver called PCSK9. And PCSK9 is really interesting because it actually sequesters the LDL receptors in the liver. It traps them and prevents them from going back to the membrane to pick up LDL and return it to the liver. So normally the LDL particles, they circulate, they deliver stuff, they get smaller, and then they come back into the liver and they get recycled, right? But when that enzyme, PCSK9, that protein protects, traps the receptors so they can't return the cholesterol to the liver. And so the inhibitor prevents you from trapping, so the liver then can return the cholesterol. And the liver piles up with this cholesterol, which I think is going to cause fatty liver disease because it can't sulfate it. And the interesting thing is that when that PCSK9 is sulfated, it doesn't trap the LDL. So what it is it's an indicator of sulfate deficiency in the liver. When there's sulfate deficiency in the liver, it can't return the cholesterol because it can't sulfate the cholesterol. If it can't sulfate, it has to store it. It has to store it in combination with fats, and that produces the fatty liver disease.
Right.
So PCSK9 is a protector of the liver, which allows the LDL to stay in circulation. You have those small, dense LDL particles circulating that are a problem. They are an indicator of sulfate deficiency in the liver, of inability to add sulfate to cholesterol. Does that make sense?
Yeah. Well, I mean, because I'm still thinking about cardiovascular disease in this mess, because you were the first person who said, well, why would cholesterol build up in the coronary arteries of all places?
I know it's the most ridiculous place to put the cholesterol, right? But there's a reason for it, because it's just like the liver stores fat, it stores cholesterol, and the heart needs to store cholesterol too, because cholesterol is so important to the heart. The heart actually really needs cholesterol to be healthy, and it needs sulfate to be healthy. And both are deficient because cholesterol sulfate's not working. Cholesterol sulfate's delivering both cholesterol and sulfate to the heart. And so when the cholesterol sulfation system is broken and those two are separated, now you've got a problem with cholesterol, non-water soluble, in those LDL particles piling up in the arteries that supply the heart, like a squirrel saving nuts. Yeah. That cholesterol is waiting to become cholesterol sulfate. And the way it can become cholesterol sulfate is by oxidizing homocysteine thiolactone. And homocysteine, high elevated homocysteine, is a very high risk factor for heart disease, worse than LDL. Elevated homocysteine in the blood.
What you're saying is that the reason it's a risk factor is because your body's trying badly to sulfate this cholesterol, but it can't. So as a marker, it's showing that you're going to have a heart disease issue.
Right. So the cholesterol is hiding in the liver arteries, and the homocysteine is circulating in the blood. It's a homocysteine thiolactone, which is a modified form of homocysteine. That molecule can be oxidized and turned into sulfate if you have reactive oxygen. So now you've got this inflammation, right? The inflammation, that's a characteristic feature of heart disease, is going to allow the homocysteine thiolactone to be turned into sulfate. And then that sulfate can attach to the cholesterol and it can take it out. And it can deliver the cholesterol sulfate to the heart. The first place it goes is to the heart. So the heart is retaining cholesterol in its arteries, in the major arteries that supply it. because it desperately needs cholesterol sulfate, but it can't get it because there's no sulfate. There's no ability to put the sulfate on the cholesterol. But if you can get the homocysteine to turn into sulfate, then you can solve the problem. And the cholesterol is waiting for that opportunity to be released as cholesterol sulfate and to solve both of those deficiency problems for the heart.
So all these years later, you still believe that the atheroma, the foam cells, all this stuff, you still believe that your body is really, this is a reaction, it's a damage. Maybe the glycocalyx was badly damaged, and this is basically scarring over. But not only that, the chemical reactions there are all being produced to try and save you.
Exactly.
Okay. All right. Well, and so, none of this looks good for big pharma wanting to study cholesterol statins, but I tell all my patients, test your vitamin D, don't test your cholesterol. If you're eating this ideal diet, a ketogenic diet, grass-fed, organic, intermittent fasting, if you're doing all these things, what are you going to check your cholesterol for? Do you really want to take a statin? Do you really want to interfere with the synthesis of cholesterol? So not good, not news for big pharma, but it's not very sexy. A cholesterol sulfate deficiency at the root cause of a lot of issues. It's not very sexy. But I think we talked about this a long time ago. Maybe even be involved in skin cancer, because cholesterol sulfate itself is one of the barrier techniques that your skin uses to protect you from radiation.
Yes.
So, because
that's also going to be deficient, and so you're going to be in trouble there as well.
A massive increase in skin cancer. Because, and I this is what I think. I think that sunscreen causes cancer.
I do too. It's actually a very good match between the rise in the use of sunscreen and the rise in skin cancer.
Yep. Well, imagine the same rise of the use of sunscreen is happening at the increase of glyphosate.
I know, they're all corresponding. And the sunscreen is an attempt to protect from the skin cancer that's caused by the glyphosate, probably,
Yeah. All right. So, to anyone who's still sticking with the details, it's good. Ultimately, someone like me, I'm going to get shouted down a lot because I'm not a medical doctor or whatever. I'm here to try and take this really cool biology and complex science and make it into something that people can get their mind around. And that's why I've got these cartoon characters. And someone like yourself who has such a profound knowledge and dismisses, you're not accepting the status quo. You're looking at the actual biochemistry and now the quantum biology aspects of, we need more people like you. We need more people like you. And I always wonder, do you ever get worried that Monsanto's got it in for you, or that?
I do. Yeah.
We had a discussion. I like to bring this up because you were instrumental in working on talk to text. And I told you this one time, and I said glyphosate, and I was alone. It was dark at night. I said, I was dictating an email to you, and I said glyphosate, and it wrote out the life of Satan.
Life of Satan. That's so perfect.
It's perfect, right? Because this molecule is the undoing of our biology.
It is so demonic. And it just frustrates me that the government refuses to accept the fact that it's toxic, and they think they can't live without it. They think we can't grow food without glyphosate. That is so ridiculous. It's destroying the soil. It's destroying the earthworms. It's destroying the butterflies and the bees, and it's destroying us.
So what do you think about? So we're talking about global warming, and if all the grasslands around the world that used to be grasslands are now farmland, which is now GMO monocrops, and it's a lot of area, North America and Europe. That's a lot. Do you think that the glyphosate could be also contributing to, if global warming were real, and I don't know.
You're not sure if it's real or not. I would say glyphosate's a major contributor, and I think it's an overlooked contributor. And I'll tell you why, because it disrupts photosynthesis. There's an enzyme called rubisco that's made by plants. Rubisco, it stands for something. The ru and the bis and the co are all, they have bigger words, but I don't know what they are. Wow. Rubisco, it's a lovely name. And that is the most common enzyme in the world. Wow. It's only produced in plants, and it plays an essential role in photosynthesis, and it's suppressed by glyphosate. Multiple papers have shown that it's suppressed by glyphosate. So this means that the plants around the world that are exposed to glyphosate cannot easily convert carbon dioxide into organic matter. And you know where that's going, because that means they can't remove the carbon dioxide from the air as efficiently as they could if they didn't have glyphosate interfering with it. And I think the plants play a major role in grabbing carbon dioxide out of the air, putting it into the soil. And eventually it becomes coal after a long time. But you're trapping the carbon in the soil and you're removing carbon dioxide from the air. Now you also have the methane problem. The cows spewing out all this methane because they can't grab the gas in time because their enzymes are broken by glyphosate. So you've got both, and we've also got nitrous oxide. That's another one that's caused by glyphosate because it disrupts the nitrate fixing bacteria in the blood, in the soil. The nitrate fixing bacteria are really important for supplying nitrogen to the crop, to the plants. And we put on all these nitrate fertilizers, and the plants can't take them up as well because of the glyphosate. And then the nitrates wash off into the waterways, and then they become nitrous oxide in the anoxic environment, the hypoxic environment in the water. They become nitrous oxide, a gas that gets released into the air. Nitrous oxide, methane gas is like 30 times as bad as carbon dioxide, as a greenhouse gas. Nitrous oxide is more like 100 times as bad.
Really?
Yeah, and so all of those are being increased by glyphosate because of the way glyphosate's disrupting these enzymes.
All right. And again, we think, to our knowledge, glyphosate is still increasing globally. We're shipping it out from the US around the world, pretty much.
Yeah, China's, I think a lot of it's being made in China now.
China. Yeah. Yep. Good times. All right. Well, that's a lot of depressing information.
We need to get rid of glyphosate. I tell you, we would get a lot further, a lot faster with our health crisis if we got rid of glyphosate. I'm very convinced of that. Of course, we got a bunch of other herbicides that are probably equally dangerous, but
you think so? All
which
right.
we'll just switch over to if we do get rid of glyphosate. They'll say, well, we can just bring back glufosinate and 2,4-D and dicamba and all these other guys, you know.
All right. All right.
Atrazine. I mean, atrazine is really bad. It's causing male frogs to become females, you know.
Wow. All right. Depressing. Okay. So I want to just make sure that you have, if anything else you want to, as we conclude, talk about, deuterium, because again, this was your, I wanted, this is actually a really good place to go to, is neurodegenerative disease.
Oh my, this will be a whole new topic. Let's do it in the next podcast.
All right.
I can tell you briefly though.
Let's have the brief.
I am actually so involved right now. In the last week, I've been obsessed with reading about neurodegenerative disease and how I can link it to deuterium. And I am getting somewhere with that. So it's really, really fascinating. I don't have the whole story yet, but I'm really sure that deuterium is playing a major role in neurodegenerative disease.
Right. And again, that makes sense. And it's funny because all these things really come together. Because when I started to do my research, I had this idea that there'd be nobody out there who had dementia of any kind or Parkinson's who also didn't already have sleep apnea. So the broken sleep was contributing to whatever was going on in the brain. And I still believe that. All the scientific studies support that. It doesn't matter what your dementia is. You're going to have probably low vitamin D as well, but definitely have sleep apnea as a syndrome, choking in your sleep and all the consequences. Okay. Well, so we're going to live to get together again. I'd love to do more of a focus on neurodegenerative disease. And
that would be delightful. I have a lot to say and I'm still working it out. I mean, it takes a while to absorb all these papers. There's a ton of papers that I'm rummaging through trying to get the story straight, but I'm getting somewhere with it and I'm quite excited about it.
Excellent. Okay, great. Well, on that note, I want to say thank you so much. We'll talk again. And I love your information. I love the way you present it. I want you just to keep on thinking like you're thinking. Keep on doing what you're doing.
Thank you. This was great. I appreciate it.
Thank you.